I need to make a correction that I think is actually more fundamental than most of the others I've walked you through across this entire series. I've spent multiple videos telling you that muscular strength predicts longevity, that grip strength outperforms blood pressure as a mortality predictor, that power specifically, not just maximal strength, is what declines first and matters most for falls and functional independence. All of that remains true, and I'm not walking any of it back. But underlying every one of those conversations was an implicit assumption I never fully examined out loud, that muscle matters primarily because of what it does mechanically, because of the force it can generate. I've been talking about muscle almost entirely as a machine for producing movement. What I think I got wrong, or at least dramatically underemphasized, is that muscle isn't simply a mechanical tissue that happens to correlate with health outcomes, because stronger people tend to be healthier in some general sense. Muscle is an active signaling organ, functionally comparable in some ways to your liver or your thyroid, constantly releasing molecules into your bloodstream that talk directly to your brain, your immune system, your fat tissue, and nearly every other organ in your body. I want to spend this entire video correcting that oversight, walking through what muscle actually does biochemically, independent of the force it produces, and why I think this reframing matters enormously for how you should think about resistance training. Let's start with the concept itself, because I think giving it a proper name helps make it concrete. Skeletal muscle is now formally recognized in the research literature as an endocrine organ, meaning it secretes signaling molecules, collectively called myokines, directly into circulation in response to muscular contraction. And these myokines then travel throughout the body and exert genuine, measurable effects on distant tissues, exactly the same basic principle by which we understand hormones released from more traditionally recognized endocrine organs like the thyroid or the pancreas. Citation. This formal characterization of skeletal muscle as a genuine endocrine organ secreting myokines with systemic effects was substantially established through research led by Bente Pedersen and colleagues, published extensively in journals, including the Journal of Applied Physiology and Nature Reviews Endocrinology. This isn't a fringe or speculative reframing. It's become a well-established mainstream area of exercise physiology and endocrinology research over the past 2 decades. And I think it deserves considerably more attention in popular fitness content than it typically receives, since almost all mainstream discussion of muscle, including a lot of my own previous videos, focuses almost entirely on force production and body composition, while this entire signaling dimension gets treated as a footnote at best. Let me walk through some of the specific myokines that have been identified and studied, because I think the actual molecular detail here makes this reframing considerably more concrete and compelling than simply asserting that muscle is more than just strength in the abstract. Interleukin 6, often abbreviated IL-6, was one of the first myokines identified and remains one of the most extensively studied. Now, I want to address something potentially confusing here immediately. IL-6 is also well known as a pro-inflammatory cytokine involved in chronic disease and the inflammaging process we discussed in a previous video. And this dual identity genuinely confused researchers for some time. What's become clear through subsequent research is that IL-6, released acutely from contracting muscle during exercise, behaves in a genuinely different, largely anti-inflammatory and metabolically beneficial way compared to the chronic low-grade IL-6 elevation associated with obesity and chronic disease. Citation. This distinction between the acute, exercise-induced, metabolically beneficial release of muscle-derived IL-6 and the chronic pathological elevation associated with obesity and inflammatory disease has been established extensively in exercise immunology research, published in journals including Physiological Reviews and the Journal of Physiology. Exercise-induced IL-6 specifically appears to improve insulin sensitivity, promote fat oxidation, and exert genuine anti-inflammatory effects through downstream signaling, essentially the same molecule behaving in opposite metabolic directions depending on the context and pattern of its release, which is a genuinely fascinating, if somewhat counterintuitive, piece of biology. Let me bring in irisin next, since I think it's one of the more genuinely compelling examples of muscle organ crosstalk in this entire body of research. Irisin is a myokine released from muscle, specifically in response to exercise. And research has found that it can travel to fat tissue and promote what's called browning of white adipose tissue, essentially inducing white fat cells to take on some characteristics of brown fat, a more metabolically active fat tissue type, specifically capable of burning energy to generate heat, citation, Bostrom and colleagues. This discovery of irisin and its fat browning effects was originally published in Nature, and subsequent research has continued to characterize its broader metabolic effects, published in journals including Cell Metabolism. Beyond its effects on fat tissue, irisin has also been found in research to cross the blood-brain barrier and exert neuroprotective effects, including some research specifically linking exercise-induced irisin release to improve memory and reduce markers associated with neurodegenerative processes in animal models, citation. This research on irisin's neuroprotective effects and its potential role in mediating exercise's cognitive benefits has been published in journals including Nature, Medicine, and Cell Metabolism. Though I want to be honest that a substantial portion of this specific neuroprotective research remains at the animal model stage, with human confirmatory research still actively ongoing and representing a genuinely exciting but not yet fully established area of the broader myokine literature. I want to bring in brain-derived neurotrophic factor here as well. Since we've referenced BDNF in previous videos regarding exercise and cognitive function, but I don't think I've properly credited muscle's specific role in this pathway before. While BDNF is most classically associated with the brain itself, research has found that exercising skeletal muscle also produces and releases BDNF directly, contributing to circulating BDNF levels and potentially playing a role in the well-established relationship between exercise and improved cognitive function and mood that we've discussed across multiple previous videos on this channel. Citation: this muscle-derived contribution to circulating BDNF and its potential role in mediating some of exercise's cognitive benefits has been examined in exercise neuroscience research published in journals, including the Journal of Physiology. I think this is genuinely important because in our previous videos discussing exercise's cognitive and mood benefits, I've generally framed the mechanism as happening primarily within the brain itself, improved cerebral blood flow, direct neuroplasticity effects. What this myokine research adds is a genuinely distinct complementary pathway. Your contracting muscle tissue itself is directly contributing signaling molecules that support brain function, meaning the muscle isn't simply an indirect beneficiary of or bystander to exercise's brain benefits. It's an active, direct participant in producing them. If you want the complete structured breakdown of exactly how muscle signaling functions connect to everything we've covered throughout this entire longevity series, and a comprehensive framework for training with this fuller understanding of what muscle actually does in mind, I put together a couple of ebooks that walk through this in a lot more practical detail than I can cover here. They're linked down in the description below. And if this reframing of muscle as an active signaling organ, rather than simply a force-generating machine, genuinely shifts how you think about your own training, I'd honestly encourage you to go check them out. Let me talk about myostatin now, since this represents a genuinely important piece of this puzzle and one that works in the opposite direction from the myokines we've discussed so far, a molecule that actually restrains rather than promotes muscle growth. Myostatin is produced by muscle tissue itself and functions as a negative regulator of muscle growth, essentially acting as a built-in brake preventing unlimited muscle accumulation. And research has found that individuals and animals with genetic mutations resulting in reduced myostatin function show dramatically increased muscle mass, citation. This myostatin pathway and its role as a negative regulator of muscle growth was established through foundational research published in Nature and has become an active area of research for potential therapeutic applications in conditions involving severe muscle wasting. What's genuinely relevant to our conversation here is that resistance training itself appears to reduce myostatin expression, essentially releasing some of this built-in brake and allowing for greater muscle growth in response to training. And circulating myostatin levels have also been found in some research to correlate with broader measures of frailty and functional decline in aging populations independent of muscle mass alone. Citation: this relationship between resistance training and reduced myostatin expression, and the broader association between circulating myostatin and frailty measures in aging populations, has been examined in geroscience and exercise physiology research, published in journals including Aging Cell and the Journals of Gerontology. Let me bring in Dacorin here as well, since it represents a genuinely interesting, if less widely known, myokine that connects to themes we've discussed regarding muscle fibrosis and tissue quality. Dacorin is released from exercising muscle and appears to work partly by counteracting some of the effects of myostatin, essentially functioning as part of a broader signaling network that promotes muscle growth and repair, specifically in response to the mechanical loading of resistance training. Citation: this Dacorin myokine and its role in muscle growth signaling, partly through myostatin antagonizing effects, has been examined in exercise physiology research published in journals including the American Journal of Physiology. I bring this up specifically because I think it illustrates something genuinely important about the broader myokine research landscape. Muscle doesn't simply respond to training through a single signaling pathway. It involves a genuinely complex, interconnected network of molecules, some promoting growth and adaptation, some restraining it, with resistance training specifically appearing to shift this entire network in a favorable direction beyond simply the mechanical tension stimulus we've discussed extensively in our hypertrophy-focused videos. Let me address apelin now, since it connects directly to something we discussed in our video on age-related muscle loss and provides a genuinely interesting mechanistic link. Apelin is a myokine that appears to play a role in supporting mitochondrial function and muscle regeneration capacity, and research has found that apelin levels tend to decline with age, with some studies specifically examining apelin supplementation or apelin pathway activation in aging animal models, finding improvements in muscle mass, strength, and exercise capacity. Citation: this apelin myokine research, including its age-related decline and its role in muscle regeneration and mitochondrial function, has been published in research including work by Vinal and colleagues in Nature Medicine, examining apelin's therapeutic potential for age-related muscle decline. I want to be appropriately cautious here, since much of this specific apelin research remains at earlier stages, primarily in animal models rather than extensively validated human trials. But I think it represents a genuinely interesting example of researchers now actively working to understand and potentially therapeutically target the specific myokine signaling changes that occur with age, rather than simply treating sarcopenia as an inevitable, mechanistically opaque consequence of aging. Let me bring the conversation to muscle's relationship with the immune system now, since this connects to something we touched on briefly in earlier videos but deserves much fuller treatment here. Given everything we now understand about muscle's active signaling role, we discussed in a previous video how amino acids released from skeletal muscle during periods of illness serve as a critical resource for immune cell proliferation. But the myokine research adds a genuinely important additional layer beyond this simple raw material framing. Research has found that regular exercise and the myokine release associated with it appears to improve immune surveillance, meaning the immune system's capacity to detect and respond to threats, including infections, and, in some research, cancerous cells specifically. Citation: this relationship between regular exercise, myokine signaling, and improved immune surveillance has been examined in exercise immunology research published in journals, including Nature Reviews Immunology. I want to bring in cancer research specifically here, since I think this represents one of the more genuinely striking, and I'll admit somewhat surprising to me when I first encountered it in the literature, areas of this broader myokine research field. Some research has found that myokines released during exercise, including specific molecules like oncostatin M, appear to have direct, measurable effects on cancer cell behavior in laboratory studies, including effects on cell proliferation and, in some research, on the cancer cells' susceptibility to being recognized and attacked by immune cells. Citation: Pedersen and colleagues. This research examining specific myokines and their direct effects on cancer cell behavior in laboratory settings has been published in Cell Metabolism and represents an active rapidly developing area of exercise oncology research. I want to be genuinely careful and precise about what this research does and doesn't establish. This is not evidence that exercise cures cancer or should replace established medical treatment in any way, and I don't want anything in this video to be misinterpreted in that direction. What this research does suggest, and what connects directly to the broader theme of this entire video, is that the relationship between muscle and cancer outcomes we discussed in a previous video, where higher muscle mass predicts better tolerance of chemotherapy and better surgical outcomes, likely reflects more than simply muscle serving as a passive reserve of amino acids and metabolic resilience. It may also reflect genuine active myokine signaling, contributing directly to a more favorable tumor immune environment, an entirely different additional mechanism beyond the passive reserve framing I've relied on in previous videos. Let me talk about muscle's relationship to bone tissue here as well, since we've discussed bone health in previous videos, primarily through the lens of mechanical loading, resistance training, physically stressing bone, and stimulating osteoblast activity. But the myokine research adds a genuinely important complementary chemical signaling dimension to this relationship that I don't think I've properly credited before. Research has found that muscle releases specific myokines, including a molecule called osteoglycin, among others, that appear to directly signal to bone tissue and promote bone formation, meaning the muscle-bone relationship isn't purely mechanical. Muscle physically pulling on bone through tendons during movement. It also involves a genuine biochemical signaling conversation happening between these two tissues. Citation: this biochemical myokine signaling between muscle and bone tissue, distinct from purely mechanical loading effects, has been examined in musculoskeletal research on muscle-bone crosstalk, published in journals including the Journal of Bone and Mineral Research. This is genuinely important additional context for anyone thinking about bone health specifically. The muscle-bone relationship we discussed in previous videos operates through at least two genuinely distinct pathways: the mechanical loading pathway we emphasized before, and this chemical signaling pathway that I hadn't properly credited, both contributing to why resistance training specifically supports bone health so effectively beyond either mechanism operating in isolation. Let me address something about fatty liver disease here as well, since I think this represents a genuinely underappreciated connection worth bringing into this conversation. Non-alcoholic fatty liver disease has become an increasingly common condition, and research has found associations between low muscle mass specifically and increased risk and severity of fatty liver disease, with some research specifically implicating myokine signaling alongside the broader metabolic effects of muscle tissue we discussed in previous videos regarding glucose disposal as a contributing mechanistic pathway. Citation: this relationship between muscle mass, myokine signaling, and fatty liver disease risk and severity has been examined in hepatology and metabolic research, published in journals including the Journal of Hepatology and Liver International. This adds yet another organ system to the growing list we've discussed throughout this video: fat tissue, brain, immune system, bone, and now liver, all appearing to receive genuine, direct signaling input from your skeletal muscle tissue, well beyond the purely mechanical, force-generating role I've emphasized in previous videos on this channel. Let me spend some time now specifically addressing why I think this reframing genuinely matters practically, rather than simply being an interesting piece of additional biological trivia layered on top of training advice that remains fundamentally unchanged. If muscle's value were purely mechanical, purely about force production, and the downstream functional benefits of that force production falls prevention, mobility, the specific practical implications would be relatively straightforward. Train for strength and power, as we've discussed extensively in previous videos, and the benefits follow. But if muscle is genuinely functioning as an active endocrine organ with meaningfully different myokine release patterns, potentially associated with different types, intensities, and volumes of training, this raises a genuinely important and I think underexplored question: are all forms of resistance training equally effective at producing this broader signaling benefit? Or might there be meaningful differences in the myokine response to different training approaches that current fitness advice, mine included, hasn't adequately addressed? Let me walk through what the current research suggests about this question, because I think it's genuinely one of the more actively developing, less fully settled areas within this broader myokine field. Research examining myokine release across different training modalities has generally found that both resistance training and aerobic exercise stimulate meaningful myokine release, though with some evidence of differences in the specific pattern and magnitude of release, depending on training type, intensity, and duration. Citation: this comparative research on myokine release patterns across different exercise modalities has been examined in exercise physiology research published in journals, including the Journal of Applied Physiology and Frontiers in Physiology, though I want to be honest that this represents a genuinely active, still developing area of research without the kind of extensively validated, precise dose-response relationships we've been able to discuss for outcomes like hypertrophy or VO2, max improvement in previous videos in this series. What does seem reasonably well supported, even amid this ongoing research development, is that both consistency and total training volume matter meaningfully for sustained favorable myokine signaling patterns, meaning this isn't an area where a single occasional workout produces some kind of dramatically different or superior biochemical outcome compared to your regular training. It's the consistent, repeated stimulus of regular exercise across weeks, months, and years that appears to produce the sustained shifts in baseline myokine and inflammatory signaling that connect to the broader health outcomes we've discussed throughout this entire video. Citation: this general principle regarding the importance of training consistency for sustained favorable myokine and inflammatory signaling profiles as opposed to acute single session effects alone has been discussed in exercise immunology and geroscience research examining long-term training adaptation. This is genuinely reassuring practical context. It means the specific training approaches we've discussed extensively throughout this entire series, consistent resistance training, addressing strength, hypertrophy, and power, combined with regular cardiovascular exercise, already appear to be producing this broader signaling benefit as a byproduct of the training you're likely already doing if you've been following along with this channel, rather than requiring some entirely separate additional training protocol specifically targeting myokine optimization that we haven't yet discussed. Let me address something about muscle loss specifically now, connecting this entire signaling conversation back to the sarcopenia discussion we've had extensively throughout this series. If muscle is genuinely functioning as an active endocrine organ, then age-related muscle loss isn't simply a mechanical problem, reduced force production capacity. It represents a genuine active loss of endocrine signaling capacity as well, meaning the aging body isn't just becoming physically weaker, it's losing access to an entire signaling network that appears to support metabolic health, immune function, bone health, cognitive function, and liver health simultaneously. Citation: this reframing of sarcopenia as involving genuine loss of endocrine signaling capacity beyond purely mechanical force production loss has been discussed in geroscience literature on skeletal muscle as an endocrine organ in the context of aging, published in reviews in journals including Nature Reviews Endocrinology. I think this genuinely changes the emotional and motivational weight of everything we've discussed throughout this entire longevity series about resistance training and muscle preservation. This isn't simply about staying strong enough to avoid a fall, though that matters enormously. It's about preserving an entire active signaling system that appears to be actively supporting the health of nearly every other organ system in your body simultaneously every time you contract that muscle tissue through training. Let me bring in something about muscle quality specifically here as well, since I think this deserves genuine attention beyond simply muscle quantity. The total mass we've focused on extensively in previous videos. Research has found that muscle tissue itself can vary considerably in quality independent of total mass, with factors including intramuscular fat infiltration, sometimes called myosteatosis, and mitochondrial density and function within the muscle tissue itself, meaningfully affecting both the mechanical force generating capacity and, relevant to this video specifically, the myokine producing capacity of a given amount of muscle mass. Citation: this concept of muscle quality, distinct from muscle quantity, and its independent relevance to both functional and endocrine muscle capacity has been examined in geroscience and musculoskeletal research, published in journals including the journals of gerontology and aging cell. This is genuinely important additional nuance. It means two individuals with identical measured muscle mass could plausibly differ meaningfully in both their functional strength capacity and their favorable myokine signaling capacity, depending on the quality, not just quantity, of that muscle tissue. And research has found that resistance training specifically improves muscle quality along several of these dimensions, including reduced intramuscular fat infiltration and improved mitochondrial density and function, independent of and in addition to whatever changes occur in total muscle mass itself. Citation: this improvement in muscle quality, specifically through resistance training, independent of pure mass changes, has been examined in exercise physiology research on training-induced improvements in muscle composition and mitochondrial function. Let me spend some genuine time now on mitochondrial biogenesis within muscle tissue, specifically since I think this connects directly to the myokine conversation in a way that deserves fuller treatment than I've given it in previous videos, where I discussed mitochondrial adaptation primarily in the context of cardiovascular training and VO2 max. Resistance training itself, not just aerobic training, has been found in research to produce meaningful improvements in muscle mitochondrial density and function. And given that mitochondria are directly involved in the cellular machinery producing and processing many of the myokines we've discussed throughout this video, healthier, more numerous mitochondria within muscle tissue likely support more robust, effective myokine signaling capacity as well, providing yet another mechanistic connection between the resistance training we've emphasized throughout this channel and the broader endocrine and signaling functions of muscle we're discussing in this video specifically. Citation: this relationship between resistance training-induced mitochondrial adaptations and broader muscle signaling capacity has been discussed in exercise physiology research examining the interconnection between muscle mitochondrial health and myokine production capacity, published in journals including Cell Metabolism. Let me bring this entire considerably long conversation together now into what I actually want you to walk away with, given everything we've covered. Muscle is not simply a mechanical tissue whose value lies purely in the force it can generate, however important that force generation genuinely is for the balance, fall prevention, and functional independence outcomes we've discussed extensively throughout this series. Muscle is a formally recognized, actively signaling endocrine organ, releasing myokines that directly communicate with your fat tissue, promoting favorable metabolic changes, your brain, supporting cognitive function and potentially neuroprotection, your immune system, improving surveillance and potentially contributing to more favorable outcomes in conditions including cancer, your bones, providing a genuine chemical signaling pathway alongside the mechanical loading we've discussed previously, and your liver, with implications for fatty liver disease risk specifically. This entire signaling network appears to be genuinely diminished by age-related muscle loss, meaning sarcopenia represents not just a mechanical, functional problem, but a genuine loss of active endocrine signaling capacity across multiple organ systems simultaneously. And critically, the training approaches we've discussed extensively throughout this entire series, consistent resistance training addressing strength, hypertrophy, and power, appear to already be producing these broader signaling benefits as a genuine evidence-supported byproduct, meaning everything we've built across this longevity series carries this additional, previously underemphasized layer of biological significance that I simply hadn't given proper attention to until putting this specific video together. If there's one thing I want you to take from this entire correction, it's this. When you're in the gym performing a set of squats or deadlifts, you're not simply stimulating the mechanical adaptations, strength, hypertrophy, power that we've discussed extensively throughout this channel. You're actively triggering the release of signaling molecules that are in that very moment traveling through your bloodstream and communicating directly with your brain, your immune system, your bones, your liver, and your fat tissue, actively supporting the health of each of those systems in a genuinely biochemical, measurable way that extends far beyond the simple force production framing I've relied on throughout most of this channel's previous content. Muscle isn't just what keeps you standing, catching yourself during a fall, and carrying your groceries. It's actively, continuously participating in a conversation with nearly every other organ system in your body. And that conversation, the research increasingly suggests, may be just as important for your long-term health as the mechanical strength itself. Before we wrap up, if you want the complete structured breakdown of everything we've covered across this entire longevity series, now including this fuller, more complete understanding of muscle's active signaling role, and a comprehensive training framework designed with this fuller picture in mind, all of that is laid out in considerably more depth in the ebooks I've linked in the description below. If you've made it through this entire considerably detailed correction, you clearly want the complete, thorough picture of why muscle matters, not just the simplified, purely mechanical version most fitness content presents. So go take a look. It's exactly the kind of resource built to help you take everything we've covered across this entire series and understand at the deepest level the current research allows exactly why the training you're doing matters as much as it does. Let me add several more genuinely important threads to this conversation, because there's considerably more depth in this research that deserves proper treatment. Let me go deeper into the FNDC5 irisin pathway specifically, since I only briefly touched on irisin earlier, and I think the fuller mechanistic story here is genuinely worth understanding. Irisin is actually cleaved from a larger precursor protein called FNDC5, which is expressed in muscle tissue and gets upregulated specifically through a signaling pathway involving PGC1 alpha, the same master regulator of mitochondrial biogenesis we discussed in a previous video on cardiovascular training, meaning irisin release and mitochondrial adaptation within muscle appear to be genuinely linked, occurring through overlapping signaling machinery rather than as entirely separate, independent processes. Citation, this PGC1 alpha-dependent regulation of FNDC5 expression and subsequent irisin release has been established in foundational research published in Cell and subsequent studies examining the signaling pathway in journals, including diabetes. This connects something I don't think I've properly tied together across this channel before. The cardiovascular training we discussed for VO2 max improvement and the myokine signaling we're discussing in this video aren't operating through entirely separate mechanisms. They appear to converge on shared cellular machinery within muscle tissue, meaning both training modalities likely contribute to this broader signaling picture, not resistance training alone. I want to bring in something about exocrines here as well, since this is a broader, more recently adopted term in the research literature that I think deserves mention, since it captures something the myokine framing alone doesn't fully address. Researchers have increasingly recognized that exercise-induced signaling molecules aren't released exclusively from skeletal muscle. Other tissues, including fat tissue itself, the liver, and even bone, release their own signaling molecules in response to exercise, and the broader term exocrines has emerged specifically to capture this entire multi-tissue signaling response to exercise, of which myokines represent one genuinely important but not exclusive category. Citation, this broader exocrine framework encompassing exercise-responsive signaling molecules from multiple tissues beyond skeletal muscle alone has been proposed and discussed in exercise physiology and geroscience research published in journals including Cell Metabolism and Nature Reviews Endocrinology. I mention this because I think it's honest to acknowledge that muscle, while genuinely central to this story and the specific focus of this video, represents one important node within an even broader multi-organ signaling network that exercise activates, rather than being the sole source of every exercise-induced biochemical benefit we've discussed across this channel. Let me address something about the specific relationship between myokines and depression here, since I think this adds a genuinely important additional dimension connecting to our previous video on self-talk and mental health. Beyond the BDNF pathway we discussed earlier, some research has specifically examined myokine signaling and its relationship to depressive symptoms, with certain myokines showing associations with markers of neuroinflammation that have been increasingly implicated in depression's underlying biology, suggesting exercise's well-established antidepressant effects which we've referenced across multiple previous videos, may be mediated partly through this direct muscle-to-brain signaling pathway, rather than exclusively through the more commonly discussed mechanisms like improved sleep, social engagement, and general mood-related neurotransmitter effects. Citation: this research examining specific myokine signaling and its relationship to neuroinflammation and depressive symptomatology has been published in psychoneuroimmunology research in journals including Brain Behavior and Immunity and Molecular Psychiatry. This is genuinely important because it suggests that when someone with depression is advised to exercise, a well-established evidence-based recommendation we've touched on in previous videos, the specific benefit may be considerably more directly biochemical and muscle-mediated than the more general exercise improves mood. Framing typically conveys providing a more precise, mechanistically grounded explanation for a recommendation that's often given without much explanation of the actual underlying biology. I want to spend real time now on kynurenine metabolism specifically, since I think this represents one of the more genuinely elegant, well-characterized examples of muscle-brain crosstalk in the entire myokine literature, and I don't think I've given it proper attention yet in this video. Kynurenine is a metabolite produced from the amino acid tryptophan, and under certain conditions, particularly chronic stress and inflammation, kynurenine can be converted into a neurotoxic metabolite that's been implicated in depression and other neuropsychiatric conditions. Research has found that exercising skeletal muscle produces an enzyme called kynurenine aminotransferase, which converts this potentially neurotoxic kynurenine into a different metabolite, kynurenic acid, that cannot cross the blood-brain barrier, essentially allowing trained muscle to act as a kind of metabolic filter, clearing a neurotoxic compound from circulation before it can reach and potentially harm the brain. Citation: Agudelo and colleagues. This kynurenine metabolism pathway and muscle's role in converting potentially neurotoxic kynurenine into a form unable to cross the blood-brain barrier was established in foundational research published in Cell and has become an influential model for understanding exercise's neuroprotective and antidepressant effects, specifically through muscle-mediated metabolic clearance. I find this genuinely one of the most compelling, mechanistically satisfying pieces of research in this entire myokine literature. It directly demonstrates trained muscle actively protecting the brain through a specific, well-characterized metabolic pathway, rather than through some vague, generally beneficial association. Let me address something about muscle's relationship to the gut microbiome here as well, since this represents a genuinely emerging, still developing area of research that I think deserves mention even in its earlier stages. Some research has begun examining bidirectional relationships between skeletal muscle, exercise, and gut microbiome composition, with emerging evidence suggesting that regular exercise and associated myokine signaling may influence gut microbial diversity and composition in favorable directions, while gut microbiome composition itself may in turn influence muscle protein synthesis and overall muscle health. Citation: this emerging research on bidirectional muscle-gut microbiome interactions has been discussed in exercise physiology and microbiome research published in journals including Exercise and Sport Sciences Reviews and Gut Microbes. Though I want to be genuinely clear that this represents one of the more preliminary, still developing threads within this broader research area, with considerably more research needed before the specific mechanisms and practical implications become as well established as some of the other myokine pathways we've discussed throughout this video. I want to bring in something about resistance training, specifically versus aerobic training, for particular myokine outcomes here, since I think there's some genuinely useful comparative detail worth adding beyond my earlier, more general statement that both modalities contribute to this broader signaling picture. Some research comparing resistance training and aerobic training directly has found that resistance training may produce a somewhat more pronounced acute release of certain myokines specifically associated with muscle growth and repair. Signaling, including decorin and follistatin, another myostatin antagonizing molecule, while aerobic training may produce somewhat more pronounced effects on certain metabolically focused myokines and exocrines more broadly, including some of the mitochondrial and fat oxidation-related signaling we discussed earlier. Citation: This comparative research examining differential myokine and exocrine responses to resistance versus aerobic training has been examined in exercise physiology research published in the Journal of Applied Physiology and Frontiers Physiology. This provides a genuinely useful additional piece of practical reasoning supporting something we've emphasized throughout this entire channel already, that a comprehensive training approach incorporating both resistance and cardiovascular training, rather than either exclusively, likely produces the most complete, favorable signaling profile across this broader exocrine and myokine landscape, reinforcing rather than contradicting the balanced approach we've advocated for throughout this entire longevity series. Let me talk about something related to fasting and myokine signaling here as well, since this connects to broader nutrition themes we've touched on in previous videos. Some research has examined whether exercising in a fasted state produces meaningfully different myokine responses compared to exercising in a fed state, and the current evidence here remains genuinely mixed and somewhat preliminary, with some studies finding modest differences in specific signaling markers depending on fasting status, while others find minimal meaningful difference in the broader myokine response. Citation. This research comparing myokine responses to exercise performed in fasted versus fed states remains a genuinely mixed, actively developing area, examined in studies published in journals, including the Journal of Applied Physiology and Nutrients. I mention this specifically because I want to be honest that this represents an area where popular fitness content sometimes makes considerably more confident claims than the underlying research currently supports. And I don't think the current evidence justifies dramatically restructuring your training and eating schedule around optimizing this particular signaling pathway specifically, given how much more established and consistently supported the broader principles we've discussed throughout this entire video and this entire channel remain by comparison. I want to bring in something about muscle-derived extracellular vesicles here as well, since this represents a genuinely cutting-edge, actively developing frontier within this broader field that I think deserves at least brief mention. Beyond the individual signaling proteins we've discussed throughout this video, muscle tissue also releases small membrane-bound particles called extracellular vesicles, which can carry a complex cargo of proteins, lipids, and genetic material, including microRNAs, and research has increasingly found that these vesicles themselves function as an additional genuinely sophisticated communication mechanism between exercising muscle and distant tissues, potentially carrying considerably more complex, multimolecular signaling information than any single myokine considered in isolation, citation, this research on muscle-derived extracellular vesicles and their role in exercise-induced intercellular communication has been examined in exercise physiology and cell biology research published in journals, including the Journal of Physiology and Frontiers in Physiology, representing an actively developing, genuinely sophisticated frontier within this broader field that continues to reveal additional layers of complexity in how exercising muscle communicates with the rest of the body. Let me address something about clinical applications of this research more directly now, since I think this adds useful, concrete grounding to what could otherwise feel like a purely academic, mechanistic conversation. Given everything we've discussed about myokine signaling and its relationship to metabolic health, immune function, and cognitive function, researchers have increasingly begun exploring exercise mimetics, pharmaceutical compounds specifically designed to activate some of these same beneficial signaling pathways in individuals who are genuinely unable to exercise due to severe illness, advanced age, or significant disability. Citation: this exercise, mimetic research, targeting pharmaceutical activation of exercise-responsive signaling pathways for individuals unable to exercise directly, has been discussed in geroscience and pharmacology research published in journals including Cell Metabolism and Nature Reviews Drug Discovery. I want to be genuinely clear that this research remains largely experimental and isn't something currently available as an established clinical treatment, and I don't think it should be read as any kind of substitute or shortcut for people who are genuinely capable of exercising. But I mention it because I think it offers a genuinely compelling almost backward demonstration of how seriously this signaling research is now being taken within mainstream biomedical research. Researchers are actively trying to bottle and replicate pharmaceutically the very biochemical effects that exercising muscle produces naturally, which I think underscores just how genuinely significant and multifaceted this signaling function has come to be recognized as within the broader scientific community, well beyond the simple, purely mechanical framing that dominated most popular fitness discussion, including my own previous videos. For so long, let me bring this considerably expanded, thorough conversation to its actual final close now. Everything we've added in this deeper exploration, the FNDC5, irisin pathways connection to the same mitochondrial signaling machinery we discussed in our cardiovascular training video, the broader exocrine framework recognizing muscle as one important node within a genuinely multi-organ signaling network, the kynurenine metabolism pathway demonstrating trained muscle actively filtering neurotoxic compounds before they reach the brain, the emerging and genuinely exciting research on muscle-gut microbiome interactions, the comparative myokine responses between resistance and aerobic training, reinforcing the value of a balanced training approach, and the active pharmaceutical research attempting to replicate these natural signaling effects for those unable to exercise. All of it deepens and reinforces the central correction this entire video has been built around. Muscle is not simply a mechanical tissue whose value begins and ends with force production. It is a genuine, actively signaling participant in the health of nearly every other organ system in your body, communicating through a remarkably sophisticated, multi-layered biochemical network that researchers are still actively working to fully map and understand. And every time you engage in the resistance and cardiovascular training we've discussed throughout this entire longevity series, you're not simply building strength, power, and cardiovascular capacity. You're actively participating in and supporting this entire body-wide signaling conversation in ways that extend the value of your training considerably beyond anything the purely mechanical framing I relied on throughout most of this channel's previous content ever fully captured. If you want the complete structured breakdown of everything we've covered across this entire longevity series, now including this fuller, considerably more complete understanding of muscle's active signaling role, and a comprehensive training framework designed with this fuller picture genuinely in mind, all of that is laid out in considerably more depth in the ebooks I've linked in the description below. If you've made it through this entire considerably detailed and expanded correction, you clearly want the complete, thorough understanding of why muscle matters, not the simplified, purely mechanical version that dominates most popular fitness content. So go take a look. It's exactly the kind of resource built to help you take everything we've covered across this entire series and understand at the deepest level. The current research genuinely allows exactly why the training you're doing matters as profoundly as it does. This video discusses general exercise physiology and biomedical research for informational purposes only and does not constitute personalized medical advice. The research on myokines, exocrines, and their effects on specific conditions, including depression, cancer, and neurodegenerative disease, remains an active, evolving area of science, and nothing in this video should be interpreted as suggesting exercise is a substitute for established medical treatment for any diagnosed condition, including depression or other mental health conditions, which deserve proper evaluation and treatment from a qualified healthcare professional. Consult a qualified physician regarding any specific health condition, and consider working with a qualified exercise professional to ensure safe and appropriate resistance training technique, particularly if you are new to structured exercise or have existing health conditions.